we started talking about ways to capture nitrogen. So for a lot of organisms, what you capture nitrogen is important because it's rare in the environment. For other organisms like us, at least in an industrialized world, you get too much nitrogen, and so you need to capture ammonia that's in your blood before it can cause your brain to swell, essentially. Okay, your brain gets hit first, really, with that. And so our two reactions that we've got are, you've got ammonia that we need to capture. That's going to react with glutamate to make glutamine. And we use ATP there. And so we're making glutamine, so it's a glutamine synthetase. And synthetase, as opposed to synthase, means that ATP is being used. Synthetase means ATP is being used. So in lab, we're doing, it's methyl tetrahydrofolate synthetase because ATP is being used. And that's going to give us ADP plus glutamine and it's not in my notes, but there's got to be a phosphate there as well. Okay, that's the first reaction that the ammonia gets grabbed up by. But actually, it's coupled with another reaction that's going to get that ammonia into to make a glutamate. So our glutamine then can react with an alpha ketoglutarate and transfer over the amine to make that alpha ketoglutarate into a glutamate. Okay, so here's our glutamine. That's going to react with an alpha ketoglutarate. Oops. Energy is required, so we're going to do NADPH. This place we use NADPH instead of NADH. And we're synthesizing something. So almost always when you synthesize something, you're going to be using NADPH. NADH is for capturing energy and then dumping it off into the electron transport chain to make ATP. And that's going to make us 2-glutamate. Plus NADP plus. And our enzyme here is glutamate synthase. Okay, it does not use ATP, and it's making glutamate in this case. All right, questions? We can add those two reactions together. And the overall reaction is, I would cancel my glutamines, right? So it'd be glutamate, glutamine, and I'm going to cancel a glutamate. I've got a glutamate here, right? And I've got 2 over here, so I'm going to cancel that as well. I've got the good math in the last lecture. So it's going to be ammonia plus alpha ketoglutarate plus ATP plus NADPH goes to a glutamate. plus ADP plus NADP plus, and that phosphate needs to be there too. All right. Questions on that? You need to know those, yes.
Where's this at?
So I want to say everywhere, but probably the liver. I think probably our best bet is the liver's doing most of this. It's going to sweep this sort of stuff up. The kidneys will do things that the liver does. Your muscles probably just lounge around and say it's not their job. Okay. Okay, so, all right. So that's important in terms of amino acid metabolism. We're in the amino acids chapter now, and what I want to do is point out that we have, you may have heard of essential and non-essential amino acids. I just kind of want you to know the term. I do not want you to know which is which. Right? You could know that about half of the amino acids for humans. This is for humans. It's going to be different for every organism what happens. Okay. So essential amino acids are amino acids that an organism needs, can't make on its own. Okay, the amino acids have to be obtained through diet. All right, so this is the list for humans. This is something I pulled off the internet. Sometimes you'll see these lists and they'll be slightly different depending on how somebody's defining what's essential and what's not. So for example, this one, tyrosine can be made from phenylalanine in one step. Okay, but phenylalanine is essential, so sometimes people will pull tyrosine over. There's some other ones where people, depending on what's happening, will call something essential or not essential. But generally, this is pretty close to the list. And I just want you to know what essential amino acids are. Okay, it's actually kind of useful in lab. There are places where you can use this to you can deprive an organism of something that is essential, and that it only gets it back or can make it if you have like a certain protein interaction happen. There are a bunch of different games you can play with this that can help you in the lab. We don't do any of that. But, whoops. Questions on that? Okay. All right, then I guess we're talking about tetrahydrofolates next. All right, so in lab, we're working with methynyl tetrahydrofolate synthetase, okay? And what it's about, I showed you some pictures before, is it's involved in folate metabolism. So what do these tetrahydrofolates do? Tetrahydrofolates are one carbon carriers. So they carry carbons in different oxidation states. For use, I'm going to say in metabolism. Okay, which is pretty general. They're going to take carbons. They're going to take carbons and give carbons to other molecules. Okay, you can think of them like as nature's Grignards. Okay, remember Grignards from organic? when you want to make a carbon-carbon bond to something, right? You make a Grignard. Unfortunately, for Grignards wouldn't work for nature because you have what around? Water, right? Okay. So in organic, you had to go and you had to put all your stuff in an oven, right, to keep water away. You couldn't have trace water. So this is an example of a tetrahydrofolate. This is related to, you've probably heard of folic acid, right? This is folic acid below. And folic acid is going to be made into tetrahydrofolates using an enzyme called dihydrofolate reductase. All right, so what you'll see here, they're not exactly lined up, but look at this ring here and look at this ring here. And you see how these two double bonds are gone? They've been reduced, hydrogens have been added. All right, and that takes our folic acid. We obtain this from diet. For humans, okay? We obtain this from diet, and then it gets reduced to this, and then these are going to be the one carbon carriers, okay? This is an unloaded. I'll show you that right now. Good question. So it's going to get loaded, and it gets loaded. Let's see. Do I like my color choice? No. It's going to get loaded at one of these places. So it's going to get loaded with a carbon at one of these nitrogens. Okay, there. Yes?
Since you labeled it loaded and unloaded, that we need to recognize.
You need to be able to recognize what is it? Is it loaded or unloaded? Draw another form. So either the unloaded form or your choice of a bunch of forms I'm about to show you. Okay? And so then this is it. I'm going to go ahead and shrink this briefly so you can see how big how big it is. And we saw this in my pre-lab lecture for the first lab, but you probably weren't paying much attention at the time. I mean, not that you weren't paying attention, but to the details of this because it wasn't our specific topic. So this is our series of pathways here. I'm going to go ahead and make it bigger again. And what I want to do is we'll start here. So this is our tetrahydrofolate. This is the molecule up above. We're just looking at the action part of it. We have this 5 and this 10. Remember, this is where we're going to load. So load here. So we'll load here, and one of the ways we end up loading it is that we can have this enzyme serine hydroxymethyltransferase. You do not need to know, I'm going to walk through this, you do not need to know these pathways, okay? Outside of what you need to talk about for lab, all right? But so this is unloaded. It can actually pull a carbon off of a serine to make a glycine. So this is one of the ways that your body makes glycine, okay, from serine. All right, and so when that happens, then you end up with methylene tetrahydrofolates. This is like ours, except we have a double bond here. Okay, a storage form of this is 5,10-methynyl tetrahydrofolate, which is right down here. So it's super stable. I can put it in a bottle in my freezer for a long time. I once talked to somebody who also did research on tetrahydrofolate. So how do you get your substrate? And I was like, oh, the methynyl is really stable. I just put it in the freezer. He's like, because he had a harder time. He had something else he was working on. It was much more difficult. Anyway, so we get the 5,10-methynyl tetrahydrofolates. So we've got this form right here. Okay. I'm looking in the wrong place. Give me a second. No, it's fine. This is 5-10-methyl tetrahydrofolate. So this is our substrate down here. Okay? And that substrate, this is our reaction that we're going to do. So this is the reaction catalyzed by our enzyme. This is actually the storage form down here. This is our 5-formyl tetrahydrofolate. This is what I've got this is the one I've got in a jar in the lab. Okay, so in bottle, in lab. I want you might need to write about it in your lab reports. Well, you won't, but that would be, you don't need to know any form. You need to know unbound, right? And you need to know a real loaded form. Does that make sense? Okay. So this might be a good one because it's easier. It's easy, yeah.
What do you mean by real?
Like if you go ahead and you stick, let's see, what can I stick on here that isn't real?
Just a carbon.
No, that's.
Like one of the ones with the red carbon.
Yes, yes.
Okay.
I'm having a little hard time actually coming up with an unreal one because almost everything's represented. But it has to be one of these. So this is what we have in lab, and our enzyme. So this is our storage form, and 5-10-methyl tetrahydrofolate synthetase will take this and turn it into the methyl tetrahydrofolate here, which can then be used in a bunch of different reactions. Okay, and so if we think about this, so this is storage form. And not a storage form, it's in a bottle. It's a storage form for your body. Yes?
Do we need to be able to name it or just label it as loaded or unloaded?
Just loaded or unloaded. I am looking at a tetrahydrofolate. It's loaded or it's not loaded, and I need to draw the other form. Okay? Which means you need to know the unloaded form, and then you need to know some loaded form. Okay, so we've got various forms here then. If we go up to this part, we end up with our most reduced form up here, which is involved. This is this 5-methyl tetrahydrofolate, and that's important this is going to be used in the synthesis of nucleotides. All right, so you would make that form to be able to use that for synthesis of nucleotides, and then down here, you've got something where we've got. Right here, we're converting between serine and glycine, right? That's used in the synthesis of proteins. And then if we come down here to the bottom, so we've got all these interconversions. This 10-formyl tetrahydrofolate, which is different from our 5-formyl tetrahydrofolate we're using in lab, this one is going to be also used in the synthesis of nucleotides. Questions?
Which one was the one in the storage form in the body again?
It's the 5-formyl tetrahydrofolate here at the bottom.
Oh, so it's in the body and
in lab, right. The reason why it's easy for us to use as our substrate, and the reason why I can have it for years in the fridge, is because of that. Yes? Mm-hmm. Okay, and so just thinking about this, I think I said this in the lab before, but if a woman's pregnant, one of the things that's advised is that you take a whole bunch of, not tetrahydrofolate, folic acid, that your body will make into tetrahydrofolate, because you're busy making a baby, right? So you need to make lots of carbon-carbon bonds. Yes?
So you said you get pregnant with folic acid, but why not just give them tetrahydrofolate? If you, like you said, that they were too busy making a baby.
it's not as stable. You have the enzymes to do it as well.
I have a question.
Yes.
So what do we need to know, like, say for just like a poster exam, like, do we need to be able to like recognize or like write down these molecules?
You need to be able to recognize it's a tetrahydrofolate if I give you one. And is it loaded or unloaded? Which means you need to be able to look at. So let's go up to the. Let's go up here.
So like we don't need to be able to say like this one's used for this.
No, you don't need to do that. I don't recall everything that I've asked for on I will have asked for something on the lab report directions, but wouldn't be the full thing. So this nitrogen and that nitrogen are the critical ones, right? You look at it, you say that's a tetrahydrofolate. I'm not even going to try to trick you by giving you folic acid, right? You recognize that this is the thing of all things we've looked at. Come up with what's distinct about this compared to our other cofactors you need to know for this. And then that nitrogen and that nitrogen are the important ones to look at. If nothing is attached, if the hydrogen's attached, it's unloaded. And if something else is attached, if you have so we've got 5-methyl tetrahydrofolate, right? We've got 10-formal tetrahydrofolate. We have 5-formal tetrahydrofolate. If a carbon is attached to one of those, that's a loaded form. And then for drawing your loaded form, pick whatever's easiest for you to remember. Yes?
So what's the point? Like, what does this do for the body? I know it adds the carbon.
Yeah.
But what, like?
You can't make nucleotides without it. The nucleotide pathways you're adding carbons occasionally, either as a methyl group. And the methyl group actually there's a DNA repair mechanism that actually, I didn't write it. It's important for making nucleotides. In fact, it's important for making thiamine, because you make thiamine from uracil, right? And the difference is the methyl group. That's where the methyl comes from. Okay, but there are formal groups in building up nucleotides as well that get added. Yes?
So you said if you're pregnant, take folic acid, the body will have tetrahydrofolate.
Yes, and then
you can make more nucleotides for like
the tetrahydrofolate is carrying, it doesn't get consumed. It is a cofactor that carries these carbon units. So it's going to grab carbons from someplace else. Like, we've got 2 places we're grabbing carbons if you look here. I just talked about, so a carbon can be grabbed off of serine, okay, you go there, or you can actually take formate and grab a carbon. Okay, that's formate, but carbon dioxide, you make formate from carbon dioxide. Other questions? Okay. Then let's go ahead, and one of the things I'm also going to ask you to do, and we're in the amino acid chapter, not my favorite chapter because it tends to be a whole bunch of different reactions. So we can look at various things, but I want you to be able to draw or show reactions of making 4 amino acids. Say 4. Hold on. I'm going to say 4 and maybe correct it back to 3 in a second, okay? Because I think I'm not allowing you to use glutamate. Let's say 3. Okay, so you need to be able to draw the reactions to make 4 amino acids. Keep changing my mind, huh? From ammonia, glutamate, ATP, NADPH, and intermediates from glycolysis or the citric acid cycle. Yeah, I'm going with 4. So first off, do we know how to do one of these? Well, ATP is not an amino acid.
What?
Well, we already know one of these, right? You're allowed to use ammonia, you're allowed to use glutamate, and you can make glutamine, right? There's one. You had to memorize this one anyway, because you need to know those independent. And then we've got some easy ones here. I'm going to actually introduce a new reaction type here. And this is, I don't actually know I think I've said this before, I don't really know my citric acid cycle and other intermediate, or some of those intermediates. I know what amino acids they are and how to easily make them from amino acids. Okay, so I've got my amino acids memorized. So we have an enzyme that's called a transaminase. Okay, a transaminase is an enzyme that can transfer an amine group from one molecule to another. And I'm going to say the amino group let's say the backbone, it's the one that's going to become the backbone. The backbone amino group of an amino acid to an alpha keto acid. All right, so like alpha ketoglutarate to make an amino acid. All right, so words here, I'm using the words the way I've got here. It's important because I want to make sure that you're not transferring the wrong amino group. Like if you have glutamine, right, you have two amino acids, you have two amino groups. So let's talk about that. So an important reaction in your body is that what you could have any particular amino acid, all right? So let's say I had serine. Okay, I've got serine, and I could react that with pyruvate. Okay. And that's going to have a transaminase. Now, there are a bunch of different named transaminases. We're not going to worry about them. All right, it's a transaminase. There's no energy involved here. It's a neutral energy conversion. But that amine from the serine is going to go over to the pyruvate. And it's going to give us whatever alpha keto serine is called. Okay, but it's an alpha keto acid. Plus, what's this?
Oh,
What was that?
alanine.
It's alanine. Okay. And this is a really important reaction. It actually doesn't happen as direct as this. I'll talk about that in a little bit. But this is an important reaction that happens in your liver, not your liver. The reverse reaction happens in your liver. We're going to get our pyruvate back from our amino acid. But actually, we're going to, there's some intermediary steps here. But this happens in your muscles. Your muscles are active, right? You're constantly turning over protein. For some of it gets built back into muscles, but if we need to go ahead and transfer the extra amines that are in your muscles to your liver to detoxify, what happens is your liver takes pyruvate, it sticks those extra amines on in this transfer, right? It takes amino acids that have been broken down, or proteins have been broken down. It takes the amino acids, it sticks the amine onto pyruvate into alanine. Alanine goes to your blood, and your liver picks it up and takes care of it. Right? And we will talk about that more. But that's one of the reactions that this is important for. So that's one transaminase reaction. So if I didn't draw it for you, right, as such, but I'm allowed glutamate, right? So if I just make that glutamate, there's one of my reactions. Does that make sense?
Pyruvate's not in the citric acid cycle.
What?
Pyruvate isn't in the citric acid cycle.
But it's in glycolysis. Did I not
No.
say glycolysis? Let me add it. I said it, but I didn't write it. So intermediates from the citric acid cycle and glycolysis. Thank you. All right, so another one. So we're allowed glutamate, right? So we can make alpha-ketoglutamate. So let's go ahead and use glutamate this time. We'll use glutamate to transfer. But so here's glutamate. Okay. So glutamate, what do you know any other things that are one step away and it's an amine from one of our intermediates? Do you remember any other ones from the citric acid cycle or glycolysis? How about oxaloacetate? Oxaloacetate is one amine away from aspartate. Okay. All right, so we have a transaminase. It'll have a specific name. But that amine, and the important thing here is it's this amine, okay? And it's getting transferred to this position. That's the amine. It's all these transaminases are only going to take that backbone amine. So if you've got a glutamate or glutamine, and you have that transaminase reaction occur, it is not the one on the end. I'll do an example of that in a minute. Okay, but then what we're going to end up with is alpha-ketoglutarate. And we're going to end up with aspartate. I blew aspartate. It's aspartate. Okay. So you need to know how to do, it's this amine to this amine. It's still primary, the backbone amine to the backbone amine position. So you need to know 4 reactions, right? Here's 2 more. You need to know what a transamination is, right? And so here's 2 more that are pretty easy. You just take pyruvate, make it into alanine, and you take, and you need to use glutamate because you have glutamate, and then you take oxaloacetate and you make it into aspartate. Yeah, Sophia.
Is the amino acid always replacing a double bonded O?
Yes.
Okay.
It's always a double bonded O, and it's that double bonded O.
And how do you? Oh, because it's like.
Okay. So now we have 3 of our reactions, right? You know, so you need to be able to complete transamination reactions, and these 2, you get 2 free reactions out of that based on what we already know. And then for your 4th reaction, go to your book and find one you like. Okay? There's a lot of them, and in terms of that, I don't want to bother showing them all to you or anything like that because I'm not going to make you responsible for them. There are too many. But in looking for one you like, you should find You should see that there are a lot of them. All right, let's real quick just show another one of these, but I'm going to do this with, let's do a glutamine to glutamate transaminase, or glutamine to alpha ketoglutarate, I should say. So here is glutamine. And here's alpha ketoglutarate. So that's glutamine, alpha ketoglutarate. All right, and what we're going to do is we're going to make whatever glutamine is without, I don't know what the name is, alpha keto acid. We're going to take this one, not that one. That's why I'm doing this right here, okay? So I'm going to just write, that's hot, danger, do not touch, okay? It's this one that gets transferred by a transaminase. And so I'm going to end up with an alpha keto, whatever this is. I don't know what the specific name for that one is. And then I'm going to end up with a glutamate because I'm going to add that amine. It's this one. It goes from here to there. And that's glutamine. Glutamate. Questions?
The first and last example wouldn't work on the test because they use things other than what we're allowed, right?
So, well, you're not allowed to use glutamate because you're allowed to start with glutamate. Okay? But you are allowed to use things you've made earlier. So you could, depending on what reactions you find, okay? I'm not going to do it here, but there's a reaction that takes aspartate, a single-step reaction that takes aspartate to asparagine, right? So you could use this as one of your reactions, right? So you could use glutamate and oxaloacetate to make aspartate, and then you could use that aspartate you just made to make asparagine. You made two. Does that make sense? If that's the reaction you decide to use. Yes?
So, this last example we did, it's like similar to that reaction up above where you said glutamate synthase, but it's like when you're with the amino acid metabolism, because you start with like
The last reaction, you're saying right here?
yeah, that's similar to like
It's sort of the reverse. Well, it's not exactly the reverse, but
that like we reviewed.
yeah. Let's go back and take a look at it. It's different.
Yeah, because there's any.
So what's going on in the reaction up above is it's this one. It's not a transaminase, right?
Yeah.
So this I wrote this one out here on the bottom to emphasize that transaminases take this amine and move it, not that one.
Okay.
All right. Up above, it was a glutamate synthase, but you're not allowed to synthesize glutamate because it's one of your starting materials for the 4 reactions. So we've got three reactions that you already need to know for other reasons. You need to know how transaminases work, and you need to know those 2 reactions at the top. Those are 3 of your reactions. And the 4th one, you just need to find one you like. But it has to follow the rules. It could be more than one if you want. Other questions? Okay.
Are you reckoning that we only do the one step reaction?
What was that?
Like all of these ones that we're learning, it's only one step.
Yeah. So you can do one step. You can do multiple steps if you want. I can tell you from the products you made here, there's at least one step reaction there. Okay. All right, so what I want to do now is move on and talk about nitrogen fixation. How time flies. Okay, so nitrogen in the atmosphere is super stable. And no plants have the ability to turn it, to fix it. At least higher plants. All right. Some plants actually form a symbiotic relationship with bacteria that can fix the nitrogen for them. So here, we're talking about beans, clover are examples of plants that can do this. And what they have is they have nodules in their roots here that actually contain colonies of bacteria. Let me see if I can, what's a good color we can see? Not that. Not that.
If this is nitrogen fixation,
Yes.
what was that first reaction is nitrogen cycle and they're different? Or is
It is the first reaction of the nitrogen cycle. I'm about to show the nitrogen cycle. Is that helpful, or are you asking about above?
what's different between this and what we talked about originally?
That's a good question. Give me a minute.
Also, do we need to know the type of bacteria?
No.
This is micro.
You need to know it for micro.
Who knows? Probably.
Oh,
I haven't learned it yet.
okay, so nodules that contain bacteria.
When in doubt, equal.
When in doubt, Escherichia. Yep.
Okay. What was that? Yeah, rhizomes. But okay, so we got bacteria in there, and so what they're going to do is the reaction above we had ammonia. Ammonia is already fixed. It's already available. It's reactive. Plants can use that directly. In fact, I've heard about some farmers actually do spray ammonia on their crops. I don't know how that works and doesn't drive you out of the area because ammonia is pretty toxic on its own. But what happens here is those bacteria are going to take nitrogen. So the nitrogen here is N2. So remember, N2 is our Lewis dot structure. Okay, we have a triple bond between those two nitrogens. You can spray nitrogen on a fire and it puts it out. All right, if you coat it in nitrogen. More fancy fire suppression systems use argon because it lays down low. Okay, because it's heavier than air. But the nitrogen plus 16 ATP plus some source of electrons. So I'm going to say 8 electrons. Different sources get used. And that's going to go to make 2 ammonia. So 2 ammonia, some hydrogen gas. So we must be getting some hydrogens from someplace else. 16 ATP plus 16 phosphate. So you can see it's pretty expensive. 16 ATP. We haven't seen 16 ATP straight out or anywhere, really, right? To take 1 nitrogen molecule and make 2 ammonias to fix for that. So what I want you to know from this is that it's really expensive to fix, and that there are bacteria that there's a symbiotic relationship with plants. And that's it. Okay. You don't need to know the reaction. But let's go ahead down and talk about the nitrogen cycle then. So we have, here is the nitrogen cycle. I like to think about this, I don't know if any of you raised fish, I like to think about this with like my fish tank, right? Which not all this is going on in my nitrogen, or in my fish fish tank. But what we have here is this is labeled, we have nitrogen in the air, okay, in plants can be fixed to give us ammonia essentially, right? Here it's got ammonium that's dissolved in water. And then what happens in this cycle is that, so plants can use this, right? But there are bacteria also that will take that ammonia when things break down, they have nitrogen available, they will start oxidizing it, okay? So they can go ahead and they can oxidize it to nitrite and oxidize it more to make nitrate. And then if you deprive bacteria of oxygen, it's useful for them. They can actually denitrify. They can reduce that nitrogen. Okay, BAF. So these are the sorts of things that happen. If you have a fish tank, you feed your fish food that's already fixed nitrogen. They make ammonia, and hopefully you've let your tank set up for a while, so you've got the bacteria that can turn that ammonia into nitrite and nitrate. Okay, but you have to change your water because they're never deprived of oxygen, so this can't happen. Another place that you see this is you see this if you think about waste, you think about a septic system that filters down, so filters waste down. And what happens is where you have the waste and you have ammonia in your waste, right? Okay. So for a septic system, and when there's oxygen available, the bacteria will take that, will break things down to nitrite and then to nitrate, and then it has to filter down far enough in the earth that there's no more oxygen access, and the bacteria down there, and there'll be bacteria everywhere, will then take it to nitrogen. That's how those systems work. Questions? Okay, I'm going to try to do this in the 2 minutes I have. I'm not going to have you super responsible with this, but I want to show you what's going on with some of these bacteria. Why do these bacteria do it? Because we can understand this based on what we know about the electron transport chain. So here we have bacteria, okay? These guys are nitrobacter, okay, for you micro nerds, nitrobacter. And so what we've got here is I want to look at this process and figure out why are these guys doing this? Where's our nitrate here? So here we have nitrite going to nitrate, all right? And then we have some electrons go this way and some electrons go that way, and depending on what's going on, you're either you're making at the end of your process NADH, or over here, I guess you're making water. So if we look at this process, so there's cool stuff I would love to talk about more, but I don't have time at this point. But what's the purpose of the nitrite in this reaction? It's both sets. Why are those bacteria doing it?
Yeah.
Yeah, it's an electron donor. Electrons go out of this into the system, so you can do that and this. Okay, that you're pumping protons across the membrane. Here you're letting them back in so that you can make NADH. All right, one more minute. Let me show you the reaction where we don't have oxygen. All right, so that's why you might oxidize these things, right? You'd oxidize them to get the electrons from them for an electron transport chain. One more second, real quick. Okay, so here, what's going on? So here we're dumping off electrons from NADH, right? And then we go down this chain and we're pumping protons. We've seen that before. And then here our nitrite gets made into NO, gets made into eventually nitrogen. What's going on in this part based on what we know about our other electron transport chains? It's anaerobic, so we can't. So what happens when it's aerobic at the end of your electron transport chain? Oxygen's your final electron acceptor, right? So here the nitrogen is acting as the final electron. It's more complicated, but we're making it so we end up with N2 as our final electron acceptor. So it's all stuff we've learned before. Okay, I'll let you go. But I think it's really cool how this stuff all fits in. And how there's not just one electron transport chain.
This is the exact lecture we had like a week ago.
Oh, micro?
Yeah.
Oh.
Same, yeah, same thing.